to talk about collecting sample data. So definitions. In an experiment, we apply some treatment and then proceed to observe its effects on the individuals. The individuals in experiments are called experimental units, and they are often called subjects when they are people. In an observation study, we observe and measure specific characteristics, but we don't attempt to modify the individual being studied. So you're just observing. Okay? So this is kind of an example of the differences between an observational and an experimental. So we have this ice cream and drowning. So observational study, observe past data to conclude that ice cream causes drownings, based on data showing that increases in ice cream sales are associated with increases in drowning. So as ice cream sales go up, drownings go up. Okay? The mistake is to miss the lurking variable of temperature, right? And failure to see that as temperature increases, ice cream sales increase and drowning sales increase because more people are swimming. So yes, when you're observing this, it may seem like there's this underlying cause that ice cream is causing drownings, but there's some other piece. So that's an observational study. You're just doing observations. Experiment. Conduct an experiment with one group treated with ice cream, while another group gets no ice cream. We would see that the rate of drowning victims is about the same in both groups. So ice cream consumption has no effect on drownings. Okay, so that would be an experiment. Okay, so here in these two, the experiment is clearly better than the observational study. Okay, so design of experiments, replication. Is the repetition of an experiment on more than one individual? So can you repeat this? Can you replicate it? Blinding. Blinding is used when the subject doesn't know whether he or she is receiving a treatment or placebo. So the subject doesn't know. Now there is double blinding too, where the subject and the person implementing the test or the experiment doesn't know who's getting the treatment and who is not. Okay, so there's blind, there's double blind, there's all these different venues with blinding. Randomization is used when individuals are assigned to different groups through a process of random selection. Okay, so randomization, just picking them, hence at random, to come up with those individuals. Okay, samples. So a simple random sample of N subjects is selected in such a way that every possible sample of the same size N has the same chance of being chosen, right? So if we have a group of people, everybody has the same likelihood of being chosen. Okay, so that's simple random sample. Systematic sample. So we're going to select every kth subject. So what's going to happen is we're going to kind of order the participants in some way, either alphabetically or numerically, some way. And then what we're going to do is we're going to select every third person to be in our sample. Convenience sample. It's using data that is very easy to get to. Okay, so this is convenience. It's simple for you to get the data, or it's simple for you to get that survey. It's people or things that are easy to get to. Stratified sample, subdivided population into strata or groups with the same characteristics, and then randomly sample within those strata. Okay, so you're going to break your population up into groups. Okay, so a lot of times you'll see this done by like male, female. So you're dividing that group into stratas or groups. Cluster sample, partition of the population in clusters or groups, then randomly select some clusters, then select all members of the selected clusters. So this is basically what you're doing is you're taking, so if you were looking at a city population and your cluster might be a 10 by 10 grid. So 10 block by 10 block grid, and that's your cluster. You might do, depending on how big your city is, you know, a few of those. So that's what kind of a cluster is. You're kind of grouping them in that. Okay, multi-stage sample. Multi-stage sample is collecting data by using some combination of the preceding sample methods. Okay, in a multi-stage sample design, a sample is selected in different stages, and each stage might use different methods of sampling. So you might do cluster in convenience. You might do, you know, kind of a variety of them. Observational study. In a cross-sectional study, data are observed, measured, and collected at one point in time, not over a period. So you're going to take that in a cross-sectional, you're just going to take kind of like a snapshot of it, and you're going to go from there. In a retrospective or case control study, data are collected from past time period by going back in time through examinations of records, interviews, and so on. Okay, so that's retrospective. So you're taking past data and you're looking at that. In a prospective or longitudinal or cohort study, data are collected in the future for groups that share common factors. Such groups are called cohorts. So they're collecting data based on these groups. Experiments. So this is confounding. Confounding occurs when we see some effect, but we can't identify the specific factor that caused it. So we see there might be something there, but we don't know why. So as in the ice cream and the drowning observational study, right? We saw that ice cream sales went up. We saw that drownings went up with it, but we couldn't see or identify that specific factor as to why. Experiment design. So completely randomized experimental design, assigning subjects to different treatment groups or process of random selection. Okay, so this is completely randomized. You're just, you know, everybody in the population is likely to be picked, and you're picking them out randomly. Randomized block design. A group of subjects that are similar, but block differ in ways that might affect the outcome of the experiment. Okay, so randomized block design. Matched pair designs compare two treatment groups, such as treatment and placebo, by using subjects matched in pairs that are somehow related or have similar characteristics. Rigorously controlled design, carefully assigning subjects to different treatment groups so that those given each treatment are similar in the ways that are important to the experiment. This can be extremely difficult to implement, and often we can never be sure we have accounted for all the relevant factors. Okay, so rigorously controlled, sometimes this is very difficult to do. Sampling errors. A sampling error or random sampling error occurs when the sample has been selected with a random method. There is a discrepancy between a sample result and the true population result. Such an error results from chance sample fluctuations. Okay, so this is a random error. A non-sampling error is the result of human error, okay, including such factors as wrong data entries, computing errors, questions with biased wording, false data provided by respondents, forming biased conclusions, or applying statistical methods that are not appropriate for the circumstances. Okay, so that's a non-sampling error when it's kind of a human error. Non-random sampling error is the result of using a sampling method that is not random, such as using a convenience sample or a voluntary response sample. Okay, so that's a non-random sampling error. And that's sample data.